Hi friends! So besides the protein extraction, another way that we can look at protein from our cell culture model is with fluorescent microscopy, and we can use stains or antibodies that are specific to certain proteins and use a fluorescent microscope. So with live cell imaging, we generally like to use a glass bottom well plate or wells or something with a transparent, optically approved piece of glass on the bottom that's going to give us the best resolution when we go to our fluorescent microscope. And so if you have live cell imaging, that means you want to see whatever's happening in real time. So we're going to use dyes like DAPI and others that don't hurt the cell for that duration of the experiment. There are lots of dyes that exist that are terminal dyes, meaning that the cells will die after a few hours or a few days if they've been treated with the dyes. That's going to be a dye like Hirsch or a Nexen. One live cell assay that's pretty common is a cell death assay with a Nexen 5 with FITC conjugated, which is a green dye. And normally that one is used for either flow cytometry or other techniques just to identify the number of dead cells versus live cells. And then there's also propydium iodide, which can be thrown in there too to separate apoptosis by a Nexen versus necroptosis with propydium iodide. Propydium iodide waits for the cell's nuclear membrane to break down, and then it gets inside that way, and it shows us which cells have their nucleus compromised. If you want to do fixed cell imaging, inside your well plate or your culture where you have your cover slips, at about 40 to 60% confluency of your cover slips and your cell growth, you can go ahead and take out your culture medium, wash it with PBS, and then fix the cells with paraformaldehyde, 2 to 3% paraformaldehyde, for about 15 minutes or so. A monolayer of cells will fix in 15 minutes. If you have a thicker tissue, you'd probably need to go for longer. The next step would be to block with 3% BSA for 1 hour. And then after blocking, you're going to permeabilize. So we want the antibody that we're trying to target to be able to get into our fixed cells, so we want to permeabilize with 0.01% Tritinex for 15 minutes, 30 minutes, and then after that we can wash with PBS, and then stain with our primary antibody 1 hour, wash with PBS, and then stain with our secondary antibody for about an hour, and then wash with PBS again. After your antibodies, then normally if you're going to do a counter stain, you can do so with DAPI or whatever, like nuclear stain or markers you're going to use. And then after you rinse those counter stains off, you can go ahead and mount your cover slip on your slide with a mounting reagent like Gelvital. And once it's dry, you can go ahead and view your slide, and hopefully you'll get a good color and good exposure. If you need to, you're probably going to have to go through optimizing your antibody concentrations to see which signal to noise ratio is best for you. So a lot of these steps you can do straight in your tissue culture hood. And So a lot of these steps you can do straight in your tissue culture hood, and you can just apply the fix and things into the well, and then you can collect the cover slips at the very end. But another way you can do it, and just in general, like if you're working with cover slips, or something like really tiny, you can put down a piece of parafilm on your table, your hood, whatever, and then just take a drop of whatever it is that you want to put the cover slip through, and so you can have like a series of drops if you need. And then we can take our cover slips and just dip them through the drops, and you can do that with PBS rinses and everything. So most likely with tweezers, you kind of just take your cover slip and let it hang out for however long it needs to, and then we can just take it